Hi, everyone. I'm Muriel Finkel, and on behalf of the ASG, we welcome you all to our AL Amyloidosis Treatment and Trial webinar. Our previous webinars can be viewed at the webinar section of our website, amyloidosissupport.org, and on our YouTube channel, and this webinar should be available for viewing in a few weeks as well. The response to our webinars have been overwhelmingly positive, so we will be continuing them along with the resumption of our in-person meetings, just not as often. Paula has a Baltimore Hopkins meeting coming up on March 16th, and over 130 have registered for that meeting. And we have about 100 that have registered for our March 23rd Mayo Phoenix meeting, so they're very popular as well. Information on all of our meetings and all of our webinars can be seen on our website, and also we post them to our private Facebook groups as well. Some of your questions were emailed in, and those will be answered first by our panel, and we also have some that will be typed in, some Q&A, and those will be answered live when we have time. Our presenters today are Dr. Angela Dispenzieri from the Mayo Clinic and Dr. Suzanne Lentzsch from Columbia University. Our pharma friends joining us today are John Sekab from Alexion, Spencer Guthrie from Attralus, Ingrid Sprinz from Prothena, Chetasi Talati from AbbVie, and Jonathan Wall from the University of Tennessee. There'll be a quick survey when this meeting is over, and we ask that you please take the time to fill it out. Should you have to leave early for any reason, please, fill it out when you leave. They're really important to us, and they really help us in developing our future webinars. You won't be able to post comments in the chat today, but please do check it out periodically, as Paula will post links and other information there. Paula will let you know how to save the chat for future reference. And speaking of Paula, in the control room today, we have our executive director, Paula Schmidt.
Hi, everyone. Thanks for coming.
And our special projects director, Bob Gibson.
Good afternoon, everybody.
Okay. We'll be working off our treatment chart today, and Bob will post it on your screen so you can take a picture of it or print your screen. You can feel free to email me at muriel@amyloidosissupport.org for a copy, so you don't have to wonder how to spell any of the treatments that we talked about today. You can email me with any questions, you know that. And now, to give us a brief overview before we get into some new and novel areas of treatment, is one of our favorite amyloidologists, Dr. Angela Dispenzieri from the Mayo Clinic in Rochester, Minnesota. Dr. Dispenzieri.
Hello, everybody. Welcome to a great session that we're going to have. If we could pull up that slide again, that treatment slide, or I can pull up a version of my own. But I just wanted to give a very brief overview of kind of what's happening today. Muriel already pointed out the different speakers that we're going to have, but there are a growing number of potential therapies for patients with AL, and most of them are treating the underlying plasma cell, the manufacturing site for the light chains that make the amyloid. And you can see all of those in every color pretty much, but yellow. If you look all the way to the right of the slide, that's where you're going to hear some of our pharmaceutical folks talk about these antifibril agents that will hopefully help the body resorb or dissolve the amyloid faster in folks. So there's going to be a lot of really interesting, exciting information presented. And we're going to lead off with Dr. Lentzsch, who's going to talk to us about bispecific antibodies and cellular therapies. And with that, I will just leave it there.
Thank you so much, Dr. Dispenzieri. And now we will hear from Dr. Suzanne Lentzsch from Columbia, and she will hold our hands as we venture into what's a new world for us in AL amyloidosis, the world of bispecifics, CAR-T, antifibrils. So let's take it away.
Yeah, thank you so much. Can everybody hear me and also see the screen? All good, Muriel? I hope so.
Yeah.
Alrighty, okay. So let's talk today a little bit about bispecific antibodies and beyond in light chain amyloidosis. I think it's the hottest topic right now in AL amyloidosis, especially because those bispecific antibodies are available in multiple myeloma, and somehow, we can steal a little bit for AL amyloidosis. So actually, we are treating patients with AL amyloidosis with the bispecific antibodies and beyond. So let me go back a little bit to explain what bispecifics are, why are we using it, and what does it mean? So bispecific antibodies belong to the immunotherapy. That means we try to destroy abnormal cells in amyloidosis, abnormal plasma cells. Sometimes we call it multiple myeloma cells, by engaging the immune system. And in principle, we have three approaches, therapeutic approaches, how to attack abnormal plasma cells producing light chains leading to amyloid. Those are the monoclonal antibodies. Everybody probably heard about daratumumab. It's a naked monoclonal antibody. The bispecific antibodies, I will focus on the bispecifics, and then also CAR T cells, the chimeric antigen receptor T cells. So all the treatments have been successfully used in multiple myeloma, and most of them are applied in AL amyloidosis, even without FDA approval. So we have two problems associated with stealing drugs or those new novel treatments for multiple myeloma applying in AL amyloidosis. Number one, the insurance might not pay for it. And number two, it's not treating amyloid patients is different than treating multiple myeloma patients. So the side effects you might see in multiple myeloma might not be seen in AL amyloidosis. A heart that is damaged by amyloid might react different to treatment than an undamaged heart in multiple myeloma. So it's very difficult for the doctors really to just take the drugs and apply for AL amyloidosis. So that's why I want to share some of the preliminary data with you. So let's go back and just look what types of antibodies we have in the treatment armamentarium for our patients. So first of all, we have a naked antibody. Everybody probably knows a naked antibody. It's daratumumab. That means it's an antibody that is artificially produced and binds to the multiple myeloma or plasma cell and directly destroys the plasma cell, or can attract other cells with a different part, with a constant part of the antibody, to really lead to destruction of the cells. Then number two, we have the antibody drug conjugates. That means it's a naked antibody, and attached to that naked antibody is a toxin or radioactive isotope. We call those antibody drug conjugates. An example for this is belantamab or Blenrep, was also used for a few of our amyloid patients. So the advantage is that in addition to binding to the plasma cells and destroying the plasma cells by binding, there's also a toxin attached that can help to destroy the cell. Number three, you can see the bispecific antibodies, and bispecific antibodies are constructs that bind to T cells, which are the good cells of the immune system, helping to fight tumor cells, but also viruses. And another part is binding directly to the multiple myeloma cells. And you can see that little red dot here. This is a target for the bispecific antibodies. It's called BCMA, B-cell maturating antigen. So in talking a little bit more about BCMA, when you hear this, and you might ask, what is BCMA? What does it stand for? So BCMA stands for B-cell maturating antigen. And what you see here with all those cells is the development of a mature multiple myeloma or plasma cells out of the stem cell. It starts with an immature B cell in the bone marrow, and then there is a maturation steps where cells are formed and mature first in the blood, in the lymph node, and then finally go to the bone marrow where they sit. So, and we also see that the BCMA is expressed on those cells. That means you can find that protein or special, I would say, identification on the surface of those cells when they are more mature. You don't find that in early cells. You only find that in cells that you can see already formed into memory B cells or into plasma blasts. That has an advantage. That means when we use the BCMA as an antigen where our antibodies can bind to, we spare the more immature cells and we only destroy the mature cells. That means the undifferentiated plasma cells and not the stem cells. So what are we doing with the BCMA on the surface of the plasma cell? Here you see multiple myeloma cells. I want to point out that in principle, you have abnormal plasma cells producing light chains when you have amyloidosis, but those abnormal plasma cells share a lot of features with multiple myeloma, and we also know that multiple myeloma can lead to amyloidosis. So please forgive me if I have multiple myeloma cell here. So again, those cells express BCMA on the surface as a marker, and that allows us that different immunotherapies can bind to that marker and destroy the cell. Here is an example of our bispecific T-cell engagers. You can see the T-cell comes from the immune system and can, when engaged, destroy the multiple myeloma cells. And here you can see the linker. This is a bispecific T-cell engager. That means it engages T-cells binding to the T-cells, but also to the plasma cells. Bispecific antibodies have exactly the same principle, just the structure looks different. Here you can see our antibody drug conjugates. Again, the antibody binds to the multiple myeloma cells via the BCMA, the B-cell maturating antigen, brings also a payload, that toxin with it, which is killing directly the multiple myeloma cell. And last but not least, in immunotherapy, you also have the CAR T cells. You can see here, those are modified T cells which we generate from the patient and modify the T cells that they express a receptor that directly binds to BCMA. That means all the CAR T cells carry the receptor that can be attached to the multiple myeloma cells and is killing the multiple myeloma cell. So today I would like to talk especially about the bispecific T-cell engagers, because we have two approved T-cell engagers, teclistamab and elranatamab, on the market. And I think it's probably the medication immunotherapy we have most experience with, apart from the CD38 monoclonal antibodies such as daratumumab. Again, just to repeat the principle, you can see this is a cytotoxic T-cell, the good cell destroying the tumor cells, but also viruses, part of your immune system. And then you have the abnormal plasma cell or myeloma cells producing the light chains. And the T-cell engager, again, engages the T-cells by binding to CD33 expressed on the T-cells and BCMA expressed on the multiple myeloma or abnormal plasma cell. So teclistamab is an approved bispecific antibody, and it has shown in multiple myeloma very promising response rates in very heavily pretreated multiple myeloma cells. We saw that in patients who were almost all refractory to daratumumab, we saw a very high response rate, a very good partial response rate of 60% with a CR rate, a complete remission rate. That means all abnormal protein was gone by 40%. And also the duration of the response was 18.4 months, 1.5 years, which I think is pretty good for a heavily pretreated multiple myeloma patient. Here again, it's the same data. You can see VGPR, 60% of those patients who had multiple myeloma, heavily pretreated, had a very good partial response rate with 40% of CR, a complete remission rate. So knowing this from multiple myeloma, we thought at Columbia, why not using teclistamab also in patients with AL amyloidosis? We were a little bit concerned at the beginning because we know that using the immune system to kill tumor cells can have a side effect which we call CRS, cytokine release syndrome. You can see that here, CRS. It means that patients have flu-like symptoms, high fever, the blood pressure can drop, the heart rate can go up. And you can imagine that in a pre-damaged heart, that means in patients who already have heart failure, that can be a severe complication if you really stress that heart. So we were very careful in the beginning and selected our patients. And what you can see here is a result of 5 patients treated with teclistamab. The very encouraging data is that all of those patients went into a complete remission or a very good partial remission. You can see 4 out of 5 patients had a CR. That means the abnormal protein was completely gone. 1 patient had a VGPR. I think what you also see is that those patients were heavily pretreated. Some of the patients had up to 7 prior lines of treatment. That really means we didn't know what to do with those patients, and teclistamab was kind of our last idea to bring the patient into a complete hematologic response. What was also interesting is when you look for especially the patient number 5, you see that those patients had even CAR T cells before, and also Blenrep, which is belantamab. It's a drug conjugated BCMA antibody. So that means that this patient had 2 different treatments targeting BCMA. And although this patient failed the 2 treatments targeting BCMA, the patient responded nicely to teclistamab. So very encouraging data. So what this graph shows you is in principle a patient journey. So each blue line is 1 patient. You can see we have here 7 patients. And what you see is the treatment, the length of the treatment in blue. The first patient was treated around 9 months, and then the next patient for around 5 months. And what you also see is with the red triangles, when the patient experienced a VGPR, a very good partial remission. And I think this is very encouraging that most of the patients had a VGPR in less than 1 month. So that means that really teclistamab works very fast, and that in median or in average, we see that most of the patients already have a VGPR, a very good partial remission after 1 month of treatment. This is really what we want. We want to induce a fast reduction of the light chains in our patients. So very encouraging data. So this is a patient example of the difference in the free light chains. And you can see that the patient received daraxonrasib, daratumumab, cyclophosphamide, bortezomib, and dexamethasone, our standard treatment for newly diagnosed patients, then pomalidomide, and then belantamab. That's a BCMA drug conjugated antibody. And what you also see is that the patient never really achieved a very good partial remission. That means difference in the free light chains less than 1. This is really the goal of our treatment. But when the patient received teclistamab, there was a fast reduction of the abnormal light chains. I think very encouraging data, again, that this drug works independent of our standard care of treatment. But even when you gave a prior BCMA drug conjugated antibody, teclistamab, the bispecific, is still working. So in summary, there are some data, preliminary data with 5 patients from Colombia that were treated with a single agent teclistamab so far, very promising response data. 4 out of 5 patients had a complete remission, and 1 patient a very good partial remission. The follow-up is a little bit short to really decide who had an organ response, but keep in mind, most of our patients in the relapse setting have only a hematologic progression. We do not really wait until there's organ response. So I guess that most of the patients might not even have a worsening of their organ response before they started treatment. The patients, I talked about CRS, the cytokine release syndrome. Remember, those are the flu-like symptoms when patients have high fever. We had only grade 1 CRS in 3 out of 5 patients. We had 1 patient dying from progressive cardiac deterioration, so worsening of the cardiac function, but not of the treatment. There are other results and other experiences with teclistamab in AL amyloidosis. There's a case report from Dr. Leung from the Mayo Clinic. And just yesterday, there was another publication from Europe in Blood, where they also shared the experiences with teclistamab in AL amyloidosis. Everything has a downside, and this is supportive care. We know that the bispecific antibodies, and that is teclistamab and also elranatamab from Pfizer, recently approved, can really lead to a decrease of the function of your immune system. That means that you have a much higher risk for infection. And that can include atypical infections such as we call that PCP or PGP, pneumocystis carinii, which is a fungal infection of your lungs, which we rarely see in AL amyloidosis and also in multiple myeloma. But at least in multiple myeloma, we saw those infections. So that's why our patients should have a prophylaxis for this infection. Also, shingles can occur under this treatment. So all the patients who receive teclistamab should also receive a shingle prophylaxis. Most of the patients, we give a prophylaxis with IVIG. Immunoglobulins are very often low after several lines of treatment, and teclistamab or elranatamab, the bispecifics, can further decrease the immunoglobulin. So it's important to have that prophylaxis. And of course, you should also have, when you're neutropenic prophylaxis. There is an occurrence of CRS possible, and we saw that in grade 1 of our patients. ICANS are the neurological side effects. We didn't see that in the, I would say, limited patient population, but those can be managed as the guidelines we use for multiple myeloma. So luckily, there's not only BCMA as a target on the plasma cells to treat AL amyloidosis with bispecific antibodies. I just also want to mention talquetamab, which is the latest approved bispecific antibodies from Janssen. And the principle is the same like we used for BCMA bispecific antibody. You can see this is a multiple myeloma cell, same for an abnormal plasma cell leading to AL amyloidosis. You see on the surface different markers with different names. CD38, daratumab binds to CD38. You see BCMA, teclistamab, and elranatamab bind to BCMA, but there's also GPRC5D. I don't know why they gave it this name, but GPRC5D is another interesting marker. And Janssen developed, and it's already approved, talquetamab for the treatment of multiple myeloma targeting GPRC5D. You can see same principle, a multiple myeloma cells is expressing GPRC5D, and then a bispecific antibody that binds to this target GPRC5D, but also to CD33 on T cells can lead to the engagement of T cells and to the killing, cytotoxic killing of the plasma cells by talquetamab. Very promising. And first data from multiple myeloma from the monumental trial in which talquetamab was used as a single agent showed there's a 60% VGPR, again, in heavily pretreated patients. And that's really the amazing thing, that sometimes we think there's nothing we can offer the patients, and then there's a new drug approved like talquetamab, and you see that still 60% of those patients go into a VGPR, so into a very good partial remission. So very promising. We also here at Columbia treated successfully one amyloid patient with talquetamab, and also that patient went into a complete remission within the first months of treatment. Also very encouraging data. And I think with this, I'm at the end of my presentation and happy to take any questions or be guided by Muriel, who is the master of the schedule.
Thank you so much, Dr. Lentzsch. And if we could have Dr. Dispenzieri, and then if Bob could put up the treatment chart, okay, that would be helpful so that we could, and Dr. Dispenzieri, if you could unmute yourself, and then we can have a little discussion here. Okay, so we were just discussing the blue column, for the second blue column. The first one's kind of, well, I guess that's blue too, but it's a shade of blue, let's say, okay? And so there you go. And so now, first of all, are these are obviously for people where you've tried everything, nothing works. So this is something that we're going to try for those people, what we call refractory disease, okay? And so obviously they've been tried in multiple myeloma before amyloidosis, as most of our drugs have, but are they infusion? How long do they take? Do we have any, and you say complete response? My thought is, well, how enduring is this response? So do you have any answers for me on any of that?
So maybe I go ahead, and give just some insight. So first of all, we were very concerned to start those bispecific antibodies in AL amyloidosis. I'm very honest. It was no man's land. The companies didn't try that. And we know that patients can have CRS, a cytokine release syndrome, and each patient, even for multiple myeloma, is admitted to the hospital for the first dosages. We start with a very low dose, and then we increase the dosage for teclistamab in 4 steps to make sure that the patient doesn't have any life-threatening CRS. I mentioned already flu-like symptoms, high fever, low blood pressure, and increased heart rate. Also, some of the patients need oxygen. Some of the patients had to go to the ICU in multiple myeloma. So we started that in patients who really had no other option. And I think I was surprised, and I want to be very careful and emphasize that what I showed you is those are few cases. Yeah, 5 cases, it's an experience, but I think it fits also with the experience others have, that the drug are surprisingly well tolerated in AL amyloidosis. I think part of that is patients with AL amyloidosis that do not have really a high tumor burden. That means they don't have tons of abnormal plasma cells. So maybe that's why we see less CRS or less toxicity. So the drug is given, it depends, but also given subcutaneously. I mentioned already, most of our patients with AL amyloidosis, we admit for a 10-day hospital stay. So that's a little bit cumbersome. They stay in the hospital, everything goes well, then those patients can be discharged. They receive initially teclistamab, I give it as an example, weekly, but then it can be switched to bi-weekly. So it will be a bi-weekly infusion. My prediction for the bispecific is, and Angela, please jump in here. It's a little bit, guessing that patients go into a very fast remission. I said that, less than 1 month. I don't think that we need to treat the patients with AL amyloidosis until disease progression or for a year or for 6 months. I think that those patients very often after 4 months are in complete remission and we can stop treatment. But again, this is just a guess, Muriel. I want to be very, very careful, but good tolerated and very effective in our patients. In red, if there's a question, are those medications that are not approved in black and then the blue background, the teclistamab and the elranatamab, those medications are approved. We have CAR T cells, very limited experience. We have Nexella, maybe we talk later about that. And we have talquetamab, which is a GPRC5D target, also approved. And we also have used that for AL amyloidosis.
Dr. Dispenzieri, what is your experience with these drugs?
Yeah, there's not a lot to add in terms of what's been said. I think the important thing to bring up is that there are a number of clinical trials that will be coming actually to test some of these drugs. So both the 2 red ones have upcoming trials. So the Regeneron one, the Levinas, I can't even say these words, but the REGN 4548 will be having a trial, and the AbbVie 383 will have a trial. And so looking forward to, kind of seeing in amyloid patients, because as Dr. Lentzsch already sort of alluded to, limited experience in patients, but unless we study these things, very formally, it's hard to really know, are there side effects that are unique and special to patients with amyloid? I mean, obviously, doctors with a lot of expertise like Dr. Lentzsch and other, amyloid centers, they know extra things to look at and all that, and they can do it very carefully. But until you really in a very orderly fashion, study these types of medications in patients with amyloid, until that's done, the real safety, I don't think we can fully describe. So I would say it's so exciting, to have these opportunities and these drugs. But, we want to tread gently as we go along, but it's great to have that opportunity.
Okay, we've got a couple of questions here. Can bispecific antibodies differentiate normal from tumor plasma cells?
That's an excellent question. Unfortunately not, that's the downside. And that's why I said, when you have BCMA as a target, you spare the very immature B cells. So you still have a kind of stem cell B cell. You don't kill the stem cell, but it cannot differentiate. And that's why you have an increased risk of infections, because in principle, you destroy all B cells, plasma cells, good or bad, that express BCMA. And that's why you have such a high risk of infection. That's a little bit an advantage for talquetamab, the GPRC5D, because that's more expressed on plasma cells and spares really the B cells. So that's why I would say we see a little bit less infection with the talquetamab than with
But
the
other side effects.
other side. Absolutely, Angela, and absolutely. We can fill the next hour with side effects from talquetamab, you know, weight loss, especially weight loss. Your patients stop eating. Yeah, that's a problem with talquetamab. Yeah.
Okay, and this person wants to know if this drug is given to patients of all stages or stage 3 and above.
Yeah. I feel a little bit, you know, I have to support what Dr. Dispenzieri said, you know. So we are sharing very exciting data. But I want to point out, see, we have 5 patients. We published our data with 2 other patients from Wisconsin. So 7 patients. I don't recall how many patients were treated in Europe. I think there are patients out. We have good, I would say, exciting data, you know, but we really have to be careful to say, you know, this is standard of care. And we were very careful with treating very advanced patients. So I think you can only give that kind of drug in a center where you work with cardiology. And the cardiologist, in my case, I'm so lucky. I have Dr. Maurer, who I always ask, you know, I said, Matt, what do you think? Can we give the patient teclistamab? Is the heart strong enough to really handle cytokine release syndrome? And only with his permission, I would treat those patients. So my answer is the patient should have a good organ function. The blood pressure should not be too low, and also the heart should not be too damaged. So I would not recommend this treatment really in end-stage heart failure. And also remember, if you go into a treatment that can lower your blood pressure, and your blood pressure is already systolic 90, you might run into major problems. So I would say, I would not kind of, you know, recommend in advanced stages. Angela, what do you think?
I support everything you said.
Okay, let's discuss CAR T for a few minutes, because I know that we've got some hot and heavy clinical trials going on CAR T, and we just did a meeting in LA where 2 patients had recently had CAR T therapy and were doing great when they hadn't responded to anything else. So let's have your 2 cents in on the CAR T, Dr. Dispenzieri.
Yeah, again, it's very similar in terms of with the bispecifics. The major concern is the cytokine release syndrome. So, and I could potentially, I'll add in a slide later to show what it looks like. But again, it's basically we're taking, you know, a patient's cells and they're modified so that their T cells will recognize this BCMA is currently the products that are available. And so then your cells are given back after a little bit of preparative chemotherapy. But what happens is, because you get the immune system so excited, which is a good thing because you want to kill, you know, you want it excited so it can kill the plasma cells. But if it gets really excited, you can have these very extreme cytokine release syndromes where blood pressures can drop, and there can be, you know, trouble with thinking and a lot of different types of side effects. And so they definitely have potential in this space. And the biggest series so far is out of Israel with a product that's not FDA approved. And all the patients got through, and they had very advanced disease, yet the long-term survival, or even the short-term, so there weren't deaths in the first 100 days, but when you look at within the year, a lot of those patients had died. So the details there, I don't know what to make of that exactly. It's unclear what the main issue is. So I think it is exciting. We definitely have 2 FDA-approved products in the US for multiple myeloma, and those, you know, are being sort of parlayed into the amyloid space. We will be doing at Mayo, hopefully opening this year, a CAR T study in amyloid patients. But it's again, the toxicity, that immediate toxicity when you're getting the therapy can be challenging. Now, the nice thing about CAR T is it's kind of a one and done approach. So it very much, the logistics of it feel like a stem cell transplant. You have to have your cells collected, except this time your cells are modified as opposed to what we do with the autologous stem cell transplant. Then you end up getting a much milder chemotherapy. You get your cells back. You have all your side effects in a shorter term here, like more 2 weeks kind of thing, or at least the worst of it. And then you're done. And you're monitored, and your immune system and the infection and all the rest is still an issue. But unlike with the bispecifics, which you're kind of doing either weekly or every other week, or maybe every 4 weeks or whatever, this is a one and done type of approach. But we have a lot to learn.
And Dr. Lentzsch?
Yeah, I was brave enough to give teclistamab and talquetamab to amyloid patients without prior, I would say, information. But I don't think I would be brave enough to give a CAR T cell or an amyloid patient CAR T cells outside of clinical trials. I think the level of CRS we see with CAR T cells is different. You can have much more side effects. Patients can end up at the ICU. I personally would not do it outside of a clinical trial, except there is a situation where I have a patient who has excellent organ function, is very young, and Dr. Maurer tells me the heart is as fit, as it can be. But in general, I would say CAR T cells in AL amyloidosis, especially in the US, where we have teclistamab and talquetamab, I think should only be performed within clinical trials. It's too risky. That's what I think. We need really a structure to evaluate what are the side effects and make sure that we have, safety tools implemented.
Okay, so we don't have any more questions typed in on that. Well, this is a question that can relate to anything. What is the difference between partial and complete remission? And I think, we use the word interchangeably, remission and response. So why don't we get the difference between remission and response, and the difference between partial and complete remission and partial and complete response. So we'll take you, Dr. Lentzsch, and then we'll take Dr. Dispenzieri for her 2 cents.
Yeah. So when we talk about a complete remission, that means all the light chains and AL amyloidosis are gone. There's no detection of a monoclonal protein, and the difference in the free light chains is normal, so it's kind of around 1. A very good partial remission is a reduction of the free light chains. The difference in the free light chains is less than 4 or 40 milligrams per deciliter. It depends really on the units you have. You can have milligram per liter, milligram per deciliter. So a very good partial remission, less than 4. And a complete remission, again, there's no detection of a monoclonal protein in your serum or in your blood. SPECT UPEP is negative, and the light chains are in normal range, as well as the difference is less than 2. You have slightly different, I would say, really characterizations of the free light chains. But again, for me, it's normal light chains, difference ideally around 1, but again, no SPEP and UPEP.
Okay, so we should state that the difference means you subtract you take the involved and you subtract the uninvolved light chain, and you get the difference. We hear a lot of people though that say, my ratio is normal, so why, what's going on? So why would the ratio not be looked at and the difference between the two be looked at? Dr. Dispenzieri.
So for a CR, the ratio is looked at. There's a time and a place for everything. What happens with a lot of these drugs, if any of patients have been on even daratumumab, you'll notice that the light chains will sometimes go so low, or the uninvolved, so if you're a lambda patient, that kappa will go so low that you can't even calculate a ratio anymore, because the number, they don't give you a proper number. They say less than 0.003 or something like that. And so the ratio becomes difficult to manage sometimes with these very potent therapies. So we'd like for there to be a close to normal ratio, or at least the involved being greater than the uninvolved is all sort of part of that deep response or the complete response. You know, so response is broken up into two major things. There's the hematologic response, which is the thing that we can see, you know, very quickly, right? We measure those free light chains, we measure those monoclonal proteins, and that can happen in pretty quick order. Month, 2 months, 3 months, you're seeing the numbers changing. But a patient doesn't really, quote, feel that response. They feel their organ response if they're short of breath or they have, you know, ankle swelling because of either their heart or their kidneys or whatever. And so we break that into organ response. And so there's a separate grouping of criteria there for organ response. But because the organ responses are so delayed, we focus a lot on the hematologic response. And that's what most of our chemotherapy, just everything about on the slide, except that right-hand yellow, induces, you know, its main purpose is to kill those plasma cells and get the hematologic response. Whereas eventually you will get an organ response if you have the hematologic response. The antifibril agents really don't do anything to the light chain itself or the plasma cells themselves, but they help the body heal and get rid of the amyloid if they work well from the tissues. So, you know, what is a remission and what is a response? You know, it's a semantic thing. We use response because it's a very direct measurement. A remission, some people will say a remission is if it's a complete, you know, that we have no evidence of the monoclonal protein, and so therefore you're in remission. Others will say you're in a remission even if you're in a very good partial response because you have achieved a nice place and, you know, we're expecting improvement in your overall function and stuff. But that remission is a sort of a funny word, at least for me. And I know that's what everybody will ask a patient, or a patient will ask a doctor, am I in remission, doc? Or, you know, a family member say, are you in remission? And again, I think, you know, if your disease is going in the right direction, that's a form of a remission. But it's, I don't know, response is easier because I think it's more straightforward personally. Because good things can happen even if you're not in a true remission with like no evidence of the clone or no evidence of anything. I don't know, Susan, what do you think about remission?
Yeah, I think, you know, keep it simple. It could be almost the same, you know. So remission means, you know, you're responding. Responding means you're in remission. So I wouldn't lose any sleep over this, you know. If you're in a complete remission or in a complete response, the doctor means the same, you know. So
yeah, you know, remission actually was the true was. Remission is if you think you cured somebody. That term was kind of the thing that was used. And in the plasma cell field, like amyloid or myeloma, you know, we're not so confident we're truly curing people, though I wonder some, you know, you have these patients that have been in response or remission for 20 years and you're like, you know, I maybe we have cured this person, you know, but so that's where that remission is kind of like it with intent to cure, not that our intent is not to cure, but we just aren't always sure we have that capability.
Thank you. Okay, we've got one here. Do some of these treatments require that the patient have specific markers for the treatment to work? So in other words, you know, for dairy, you need to have a CD38 or whatever. Do you need a specific marker for some of the treatments that we've been discussing here?
Nothing. Yeah, nothing. So there is some discussion whether you should measure BCMA. We know that the plasma cells can shed the BCMA, the antigen, into the bloodstream, and whether you should measure in the bloodstream or directly on the surface the expression of BCMA and only treat patients who express or have BCMA on the surface. But data have shown that that's not really necessary, and we don't do that. So we treat all the patients. I think the biggest criteria to treat a patient would be really the organ function. How good is the heart? How good is the blood pressure before I give those bispecific antibodies? So
Yes,
no,
that would
necessary.
That would fill in what this person wants to know. Is there a difference in the side effects if you have MM and AL? And obviously the heart could be one issue, right? What would be, and when you notice the side effects for these treatments that we've been discussing, what were the different side effects that our AL amyloidosis patients showed versus the multiple myeloma patients?
Yeah, maybe I should take that because we treated the patient at Columbia. I think it's really, and I have to really support what Angela said, it's really too early, and we have too few patients to have a final conclusion. From the limited number of patients we treat at Columbia, I can say the treatment was very well tolerated. We didn't see any major side effects, but it was a selected patient population, you know, patients in good shape. If I can speculate, I would speculate that maybe the patients have less infectious problems we see with multiple myeloma, because most of the amyloid patients are less heavily pre-treated and have a lower burden of tumor cells. So our myeloma patients can have a complete infiltration of the bone marrow, 90% plasma cells. We rarely see that with amyloidosis. So most of our patients have a lower tumor burden, so maybe less CRS, cytokine release syndrome, because that can also depend on the tumor burden. And I think the patients are less heavily pre-treated, so maybe we see less infectious problems. But again, Muriel, I think it's a little bit too early to really give a final statement on the side effects.
Okay, look, well, we have you, Dr. Lentzsch, for one more minute here. We didn't touch the antifibril agents, and that's our yellow column, okay, and these are the treatments that will hopefully vacuum out the amyloid from the organs and solve a lot of problems if they work, right? So I know you were in at the very ground of the KLM 101 trial, which is now called ensalutamab, whatever.
Yeah.
There, right. And so I was wondering, is that trial filled now at Columbia? And when do you think that we'll have some results on that? I know we'll be hearing from someone soon on that, but I wanted to get your take.
Yeah. So the trial filled accrual, so that we had 2 trials, one for patients with advanced heart disease stage 3A, and the other trial for 3B. And I think at the end, everybody was surprised that the trial could meet the accrual relatively fast, despite there was COVID and there was a delay in clinical trials. So I think what we are waiting right now is for the results to see whether there's a difference in the patient group that received placebo versus the patients receiving really KL 101. So I'm sure that we have the colleague from Alexion, from AstraZeneca, knowing more about the trial. But from my side, we have to wait until we have enough events that show that there's a difference in the outcome between the placebo and the treatment arm.
Okay, and Dr. Dispenzieri, I'll ask you about the NEOD-001, now known as Prothena. How's that trial? Is that trial filled and accrued, and how's that going?
That one's still accruing, and it's also going after sort of more advanced amyloid patients. They use the 2012 staging system, so it's stage 4 patients, and so that is still accepting patients, but it's a very specific cohort of patients. The eligibility or can sometimes be a wee bit challenging, but it's both of these were just newly diagnosed patients, so it's not for second line therapy at this juncture or anything like that. So
okay, well, let's take 2 questions and then we'll say thank you and go on to the next thing, because we're going over time, of course. Okay, are these drugs good for MRD positive patients? We're talking about the bispecifics and the CAR T.
Yeah, so I don't treat MRD positive patients. I'm very honest. I have patients post stem cell transplant who were MRD positive, and there was a discussion, should we give maintenance or not? I still see them, doing very well for many, many years. I think that would be too aggressive to use teclistamab or elranatamab or talquetamab to treat MRD positivity. Yeah, we don't have enough data. What's the role of MRD positivity in patients with AL amyloidosis? So again, I wouldn't do it. I don't know, Angela, how would you approach this?
yeah, totally. So just to explain what MRD is, so the idea is we have these degrees of hematologic response. And so, a partial response is like 50% better in the light chains and the VGPR, as Dr. Lentzsch had shared, and CR, again, further, less evidence. But then if you go to the bone marrow and you can still see, if you look really, really hard, still some of those plasma cells, which are the manufacturing cells for the light chains and therefore the amyloid, do you treat based on that when so much else is good? And I would agree with Dr. Lentzsch that at this juncture, I wouldn't really recommend it. I think that so many patients can do well. I mean, patients even can do well with a VGPR and not a CR, or a complete response. And so, but I think that, the more tools we have, the greedier we get, and greed sometimes can be good. But we also want to make sure that we're not hurting people by, just focusing on numbers too much. I think you have to look at the whole picture of how they're doing.
Okay, great. Thank you so much. We'll take those other questions later on. I promise we'll try and answer all our questions, but we have to move along right now. And so we're very happy to have with us Dr. Jonathan Wall from the University of Tennessee, who's an old friend. Oh, with the Stigmatism Kitchen, there we go in the background. Hi, Dr. Wall.
Thanks, Muriel. How are you?
Fine, thank you.
I appreciate the opportunity for the invitation to talk, so I shall.
Yeah, we hear you have a new study that you're recruiting for, and we want people to hear about it because they always love to get imaged.
So yeah, thank you. This is a new study. So we're very interested at the University of Tennessee in developing ways to help amyloid patients. And in addition to the antibody therapeutics that Spencer talked about, we've spent a lot of time working out how to detect amyloid early and accurately and with some sensitivity so that patients can get treated earlier and more effectively. And so as Spencer took all the thunder for the previous imaging study that we did, which was a PET study using a peptide, we've now developed another imaging agent for a different type of imaging called SPECT imaging, which is single photon emission computed tomography. And I will tell you a little bit about that and why we have something different, other options. I have the following disclosures. The most important one is that, as Spencer mentioned, I'm a co-founder with him of Attralus. So what's the agent actually look like? As again, Spencer mentioned, we developed this peptide that we called lovingly P5+14, and we radio labeled it very early on in that initial study with a type of radioactivity that you could use for PET imaging, which is a scanner that's used mostly in oncology for oncology imaging, and those scanners are in hospitals and big hospitals and academic centers. And Spencer showed some of the images, and the thing to take away from those images is the peptide was very good at imaging amyloid in the heart. And heart amyloid is the, I guess it's the most important with respect to the prognosis and how well the patients are functioning, how well the patients do. So being able to detect heart amyloid easily and accurately and early, and getting the doctors to have the ability to order a very easy scan that can rule it in or rule it out, we think is very important. So in addition to that original PET agent, we've developed this, and it's based on exactly the same peptide, which is, again, Spencer mentioned, has been used in over 130 patients now, some around the US. And it forms this really quite beautiful alpha helical shape. And the little legs on the caterpillar are the things that interact with the amyloid. And we label it in the lab with a type of radioactivity called technetium-99, which is the most commonly used imaging radioactive molecule in the US. There are almost 100 scans that are done with different scans, traces that are labeled with technetium. And throughout the US, hundreds of millions of scans are done every year with this reagent. So it's very safe, and we all know how it works. And what we do is we put the technetium on the end of the peptide, and what that allows us to do is wherever the peptide goes will be injected into the patients, is to detect it using this type of imaging by actually looking for the radioactive signal that emits from it. And the way we think that this actually binds to fibrils And again, as Spencer mentioned, we're very interested in developing things that combine all types of amyloid, AL, TTR, and all the rare forms as well. And this is one of those peptides. This is very closely related to the peptide that's used on the therapeutic ATO2 that he was mentioning. But on the right here, you can see, so this is a model of what a fibril looks like. All the sort of the gray shape is what a fibril looks like. And we think the peptide lies along the surface or the edge of the fibril as it sort of extends in and out of the screen. And that shape is common to all types of amyloid fibril, and that's how we think this peptide can image all types of amyloid. So the goal is to solve this problem that is faced by patients, and that's early and accurate diagnosis. And as Dr. Dispenzieri mentioned, the major centers, the people who see amyloid patients all the time, these guys are absolute experts at doing that. But in the community setting, in Knoxville, for example, and other places around us, these guys are not used to seeing amyloid patients, and they don't think about it. So the goal is to try and enhance the ability of those other centers to accurately detect amyloid and then move the patients into effective treatments. And that's basically said in the bottom part. And nowadays, the cardiologists are seeing a lot of amyloid patients, TTR in particular, but also AL, as well as the hematologists. And so this is a tool that we think that could be used mostly by or adopted by cardiologists in the community. So the study itself, which started late last year, is a single site. It's only happening in Knoxville. It's a multi-part study, and it's a phase 1. So this is the first time that this agent has been in people, and it's of that peptide labeled with technetium. All the patients are outpatients, so you just come to Knoxville and spend some time with us and get imaged. You have to have a confirmed diagnosis of AL or TTR amyloidosis. And right now it's within the last 2 years, but we're going to change that. So you have to be diagnosed within the last 4 years. But importantly, in terms of safety considerations, there are criteria that are specific for how well your heart is functioning and how well your liver is functioning. So we need to assess those values before you'll be eligible to be imaged. As I mentioned, this is a first time it's been done. It's a pilot study of 4 parts. The first part will be assessing the radiation dose, which is a standard thing to do whenever you do a new radioactive molecule, and we'll do that in AL patients. The second part is enrolling about 5 AL patients and 5 patients with TTR amyloidosis, just to get some early data. Part 3 is looking at the peptide and how it distributes around the body in healthy volunteers to understand what that looks like, because that's extremely important. We've completed that part of it. And part 4 is this rare or relatively rare group of TTR patients who have proven amyloid in the heart, but for some reason they do not light up in another type of scan that's used for TTR patients called PYP imaging. And so that's just for that group as well. So what does the SPECT scanner involve, and what does the trial involve? So the patients come to Knoxville, depending on what part they're in. They'll get injected with the radio tracer after we do some tests and some blood work. And then you lie on a scanner. And this is a SPECT CT scanner. And basically it's a bed that you lie on, and above you and below you are 2 gamma cameras that capture the radioactivity. And we capture 2 types of images at 2 time points. So we do what's called a planar or a 2D image, and we do a 3D image at 1 hour and 3 hours post-injection. And for the planar image, you just lie on the bed and the camera sort of scoots across you, and we get an image like you can see in the black and white picture here. So whole body distribution of the radioactivity. And then for the 3D image, the cameras are placed pretty close to your chest, and they spin around you over about 40 minutes. And we can generate 3-dimensional images. And that's the sort of the colored images you can see over here, where you can see the patient's heart light up. And so combination of those 2 images will allow us to determine where the tracer goes and how well it picks up heart amyloid, and what else we can see with this tracer in patients with AL and TTR. So we've done to date about 10 subjects, 5 healthy volunteers, and 4 patients with ATTR. We haven't had any patients with AL, so we're still excited to see what that looks like. So I'll just show you some examples of the data we've generated. And these are those 2D sort of planar images, the black and white. This is a map of radioactivity, and the dark the sort of black color is where the radioactivity is. I don't need to point out where everything is, but you can see in the healthy volunteers on the left, this is 2 of them, that there's no uptake in the heart. So there's just a light gray sort of area where the radioactivity is still in the blood, but you can't see any image of the heart in both of the healthy volunteers. And this is the heart in a patient with TTR amyloidosis at 1 hour post-injection and then at 3 hours post-injection. So the data so far, with just a very small number of individuals, but quite a few healthy volunteers, suggests that the peptide works exceptionally well for imaging the heart. And that's perhaps not surprising because I mentioned it's the same peptide that we've used in our PET study where we saw the heart beautifully in a lot of the images that we took. So this reagent is working well with this radioactivity on it. The peptide's cleared through the kidneys, so the kidneys light up in everybody, and some of it's cleared through the liver, so the liver lights up in everybody. But importantly, this is a cardiac imaging agent specifically designed for that, and it works exceptionally well so far. Again, this is TTR patients. We haven't had the opportunity to look at AL. Just to zoom in on that image, and you can see sort of this is the region where the heart would light up. This is just blood pool. This is the spleen actually over here, but there's no evidence of the heart. You can't see any structure. In a patient with TTR amyloidosis, you can see the left side of the heart and the right side of the heart in this image really quite well, with no interference from anything else in the thorax, any activity up in the chest area. Just to trump Spencer a little more, here's a nice spinny image because he doesn't have one of them in his presentation. On the left, so that's a spinny image of our peptide in a patient with TTR amyloidosis, and you can see the heart sort of spinning around up here. You can actually, as it spins round, you can actually see into the heart, into the left side of the heart, and see where the blood it would actually be. That's the resolution of the images we can get. And I've actually fused it with a pyrophosphate, a PYP image, so you can see that the two things completely overlap. And we can see what the PYP can see, but we can also see more, I think. And on the right, there's images again of the top row is our peptide in this patient, and you can see the left wall, the wall that divides the left and right side of the heart, called the interventricular septum, and this is the right side of the heart. This is one patient and 3 different views of the heart lighting up really well. Below that is what's considered to be the standard of care for TTR imaging. This is this PYP scan, which works very well if you have a lot of amyloid and the scan's done properly and it's done in an academic site or a site where they really know how to do these scans. It works very well, but in a lot of places it's a problem to do well. But you can see that in the same patient, our tracer, new tracer, lights up very well where the PYP is. This is something we've learned while doing this. This is another patient and another spinning image. This is just our peptide. And again, if you look, you can actually see down into the heart. So this is the heart, the upper blob here, both kidneys. But this shadow behind the heart is the lung. So this is what we think at this moment is lung amyloid. And if you look at the images on the right, you can see the heart uptake, and in the bottom panel, you can see that shiny area, which is sort of on the back. It's on the lower part of the image, but it's the back of the lung behind the heart. And we think now that one of the things we're learning is that not only might we be able to image cardiac amyloid exceptionally well, but we might be able to detect lung amyloid in the same image and get twice the information. Both those things are important because those are, in addition to GI amyloid, heart amyloid and lung amyloid, really impact quality of life with respect to activity and how things change in what you can achieve. So having this information would be really quite valuable. So from our early observations, as I mentioned, we've only done 5 patients and 5 healthy volunteers. We can say pretty well that the peptide does not bind amyloid-free hearts in healthy volunteers, and it does have the potential to bind ATTR amyloid. I would love to say that we can say the same thing about AL, but I think it's just a matter of time before we get some patients with AL amyloidosis to come to Knoxville and help us out. Amyloid in the lung, as I just mentioned and showed, may also be imageable with this agent quite effectively. And this is a pilot study, and our goal is it's enrolling just a few patients just to get proof of principle. But our goal is to expand it with more funding and include more AL and TTR patients to learn more about it, work on how to quantify these images, And if you'd like any more information, there's an email at the bottom here of the slide, and it's not my email because, as most of you may know or probably do know, I am absolutely useless at answering emails effectively. But Dr. Martin, who works with me, is a superstar, and she will answer your emails. So if anyone's interested, please feel free to drop us a line. And this is the group, and we're very fortunate to have the support of Muriel and a lot of other people with what we do, and the opportunity to talk to you guys about the exciting things that we're doing. And again, we couldn't do anything in our lab without your help, and we really appreciate everybody's involvement in everything we do in Knoxville.
Thank you so much. There'll be questions in a few minutes, but one comes to my mind immediately. We put this out to our AL patients, and you don't have any AL patients yet. And what is their reluctance? Is it laying for 3 hours on the?
No, it's well, you don't lay for 3 hours. It's 40 minutes, and you do get to hang out with me all day, which is a huge bonus. The issue is that heart criteria. So patients with AL amyloidosis, a small amount of amyloid can cause a lot of heart problems, and there are other reasons why. So the FDA made us set what's a QT interval limit. So the QT interval is 2 spikes on your heart EKG and how far they are apart. And if you're above 480 milliseconds, then the FDA said those patients shouldn't be imaged at this early stage. So we're trying to find patients who have heart amyloid, but not a bad heart. And that's tough. It's much tougher in the AL population.
So it'd be like a stage 2?
You would have to ask Dr. Dispenzieri what stage that would fall into. But a 1-2 would be safer than a 3-4. I imagine. Yeah. So it's not that we haven't had lots of people contact us. It's just that we've had to be very strict in this study, sadly, about filtering out who we can and cannot invite to Knoxville.
Great, thank you. We'll be doing Q&A in just a few minutes, and so stay tuned, everyone. But now, Paula Schmidt has some financial aid news for us, and she's going to take a few minutes and share that with us. So, Paula, take it away.
Good afternoon, everyone. I hope I can give you a little bit of good information today anyway. But while Bob's pulling up my slides, if you go and pull up the chat feature that you can locate down at the bottom of your screen or wherever your toolbar is floating around for the viewing area on your screen, I have already listed several websites that you can go to. I have posted our financial assistance flyer link that gives you a good overview of these financial assistance programs, as well as the percentages that they use, whether or not they have a wait list, that type thing. So be sure to check those out. And while I'm saying it right now, in order to save this chat, you want to go, if you have the window pulled up for the chat, if you go down to the bottom right-hand corner, you're going to see 3 dots out next to the little smiley face. Click on that, and it will have at the top of their little menu that pops up, Save Chat. That's all you have to do is just go to the 3 dots and then click on Save Chat, and you will have it. Just know where you saved it on your computer so you can go back and find it again later. All right, next slide, please, Bob. Currently, the Healthwell Foundation has a myeloma fund open. If you have AL and myeloma, you can possibly qualify for up to $12,000 in assistance if you are on Medicare and if you meet the 500% federal poverty guidelines, which you can find on that chart that I have put into the chat. Also, if you have cardiomyopathy and AL amyloidosis, this fund tends to stay open, I'm going to say pretty much year round. I have not seen it closed this year with the Healthwell Foundation. Go there. You have to have Medicare in order to apply for that program, and you also have to meet the 500% federal poverty guideline. But this fund, even though many of our AL drugs are covered by Part B Medicare, things that are covered by Part D, drugs that might be covered are Entresto, Farxiga, Jardiance, doxycycline, spironolactone. Those are just a few of them that I saw on the list yesterday. So if you have any out-of-pocket prescriptions where you have an out-of-pocket cost, please go to and check out the cardiomyopathy fund, see if you qualify for it. And that will help cover your drug costs, or you can also you could opt to have it cover your Medicare premiums for the year. So you can do one or the other, and you can change that once each year. But go to that website, healthwellfoundation.org, to check out those two funds. Majority of the other funds right now are closed for AL and for multiple myeloma. Next slide, please. The Leukemia and Lymphoma Society, even though I have listed on here that they pay up to $5,000 a year if you have AL and MM or AL and Waldenstrom's, both of those funds are closed right now. They do not have a wait list that you can sign up for. So you have to check back to this website often to see if it has possibly opened up. Now, they do have some funds open right now. One is a patient aid fund, which will send you basically a $100 check for having a diagnosis of myeloma. You do have to have myeloma along with this to qualify for these funds. They also have, in some areas, there is a local financial assistance program that they have that is open in a few areas. It's done basically by zip code. So you would need to go to their website and check that out to see if it is open in your area. But they use basically a 600% or below federal poverty guideline. So you can make a pretty good amount of money each year and still qualify for assistance through them if you have myeloma. Patient Advocate Foundation, their funds are closed right now as well. But some of these foundations do have wait lists that you can go in and sign up for. So do that if they have that availability. If you're on Darzalex and having problems with getting that drug paid for, that website right on the screen now at the bottom will help you get assistance for that. They also have an assistance fund, foundation fund, which is available to people with no insurance or high copays, something like that, or your insurance doesn't want to cover the drug. You can call them up and talk to them as well. Next slide. This is the threshold chart for the federal poverty guidelines for all of these different foundations. They range anywhere from, you can have 400, 500, 600% tend to be the percentages for AL amyloidosis. And let's just say you have two people in your family, you're going to go to the 2 on the left-hand side and then scroll over to 500%. So you can make $102,200 a year and still qualify for assistance. This is for people just living in the United States. All of these programs are for U.S. residents only. I'm sorry to say, I know we have people here from outside of the U.S. on this webinar, so I apologize for that, but this is just for the U.S. right now. Next slide, please. Now, to help you also know when these funds open up, you can sign up for what's known as the Fund Finder app through the PAN Foundation. If you go to fundfinder.panfoundation.org and sign up for this and put down amyloidosis, they will list all these different foundations that have programs for amyloidosis and possibly myeloma, if you put that down as well. And when I did this one, see the little green lock on the right-hand side that is open, that shows that fund is open. This is an old slide though, so it says myeloma associated amyloid was open at that time. Right now, it's just myeloma that's open. Or no, excuse me, myeloma is closed. It was open a couple of weeks ago, but that's what it will look like if a fund is open when you first sign up for this. Now, they will send you an email that alerts you to when the fund opens up. So this is a good thing for you to sign up for. You know, I said that the Leukemia and Lymphoma Society did not have a wait list. This is their wait list, basically, because you can sign up for it at Pan Foundation and they will alert you when the Leukemia and Lymphoma Society opens up. Now, this year we've had changes to our Medicare program. Medicare Part D is, you know, the books that they send us say that you can have to get up to $8,000 out of pocket before you go into catastrophic coverage, but that's changed somewhat. I cannot find it in writing anywhere exactly what the changes are. We've heard people say that their insurance company has, once they paid $3,000 out of pocket, the insurance company was covering everything else. Some of them have said $3,250. I just don't know for sure what that amount is. If you go to triagecancer.org, I also have that website listed in the chat. They have a lot of information on that website for Medicare, health insurance, anything related to health, healthcare. Go to that website and don't let the cancer word in that title scare you off. They cover many different illnesses. It's just the name of the organization, but they have wonderful information on that website and you can learn a lot from them. But I have bookmarked that as well in the chat. So just go to those, be ready to jump on these funds quickly when they do open up. If you get a notice saying it's open, apply quickly because it could close the next day. It just depends on how many funds they had available and how many people apply quickly. So you want to be ready to do that. So do it just as soon as you can when you see that it's opened up. And good luck to you. And I hope you can qualify for some of these funds. Thank you very much.
Thank you so much, Paula. Paula always ends up being everybody's best friend, and we see why, right? Okay, now if we could have Dr. Dispenzieri and all of our panelists here, we've got some questions to answer. So we'll get going on these. Let's save the DARA questions, okay? There's quite a few DARA questions, okay? But this is a question that comes up quite often, Dr. Dispenzieri, and that is myeloma and AL amyloidosis different from MM alone, or is AL myeloma less malignant in any biological way? And we do see that. We see someone say if you've got 10% plasma cells, you have myeloma. If you have 60%, you have myeloma. Some say you don't really have myeloma because you don't have CRAB. Help us here, okay?
Sure. So again, amyloid and myeloma are both what we call plasma cell disorders. So that's too many of these bone marrow plasma cells. The average or median number of bone marrow plasma cells of an amyloid patient is about 10%. So that means half of patients will have fewer than 10% of these plasma cells, and the other half of patients will have more. If you have over 10%, in theory, you can fit into a myeloma category. Most patients with AL amyloid, however, even if they have 15 or 20% plasma cells, which does shift them, you can use the word myeloma, which is convenient because you can get some of these funds and you can, get access to certain drugs when your doctor uses those words. But most of the AL patients, even with 15, 20% plasma cells, don't act like myeloma. So myeloma patients in general have pretty healthy organs. They'll have bad bones, sometimes sick kidneys, but it's really that the bone marrow is extra sick. It makes them anemic. And so it's just a very different disease. Whereas amyloid patients, their bone marrows don't look that bad. I mean, it's abnormal. You have more plasma cells than you should, but the problem is those plasma cells are they're making these amyloid proteins, these light chains that are then making the organs sick. So it is a spectrum. It is a little bit semantics. We will often use the myeloma term more in a patient's chart just to get access to different things, be it a drug, be it insurance opportunities, and so forth. The CRAB that you mentioned, so myeloma is associated with what we call CRAB, which is calcium is high, renal dysfunction, but it's a different renal dysfunction than what most of our amyloid patients have, anemia and bone disease. Fewer than 15% of amyloid patients will have, the bone disease like we see in multiple myeloma. So I hope that helps a little bit.
Oh, it's very helpful. Thank you. And here's a question that KALM 101 trial only includes newly diagnosed AL patients, correct? And we know this is true, both for both of these drugs. It's for AL patients only. So both of your trials are what they call event-driven, the event being death. And so if we substitute the word dance for death, which I did recently in a meeting, people preferred it. Okay, so what we're saying is that you want newly diagnosed patients that have very sick hearts, right? Because we do see that with AL amyloidosis, right, Dr. Dispenzieri, that those are the patients that we frequently lose that first year, are the people that have very heavy cardiac involvement. So that's where we're going to be able to show the FDA that there will be more events with those people, if but with Daraside or D, we see that they're living longer because they're going into complete response quicker. After 1 month or 2 months, they're going into complete response, which means in theory, their heart is not getting as damaged as it was in the old days. So does this mean that these trials will take a lot longer before these events take place?
Is that for me or somebody else?
That's for you.
Yeah, the good news and the bad news is patients are doing better. I mean, it's certainly the good news, but in terms of these trials and getting the answers, it will take longer to know if there is a benefit. And so, patients everybody's impatient. We want to see the results of these studies, but the better patients do, the longer it will take us to really know how good these drugs are. So it's a mixed blessing.
Right. Bob, if we could have the chart put up for some of these questions, that would be really helpful. Okay, so another patient asks, if a patient is on the KALM 101 study, are they also eligible for bertamimab? And we see that they're both in the same column. So KALM 101 and bertamimab. So in other words, they're competitive drugs in trials. So one could be on both of those anyway. They wouldn't meet the criteria. But let's say both drugs were approved. Would there be any reason for them to be on both? Dr. Dispenzieri, anyone?
In general, you don't use things that are in the same column simultaneously. Or even necessarily, if they're really similar, you're not gonna, if one hasn't worked for you, like the daratumumab, isatuximab. So if you go to the first column, the blue column, those drugs are very similar. They're naked anti-CD38 antibodies. And it's been shown that, if one stops working or it doesn't work, you probably don't have a lot of benefit in going to the other one. And I would presume if both the KAL and NEOD are effective, I think people are going to have to, choose. And sometimes it's, who made it to the market first. I think daratumumab, I don't know that it's really any better a drug than isatuximab, but they got there first. And so that's what people, that's the go-to because it's approved and that's where all the excitement is.
Okay. Someone asked clarification on Dr. Wall's new study without AL patients. Is one of the qualifiers that patients diagnosed more than 4 years ago do not qualify? So I believe you said initially, Dr. Wall, that you wanted people that were diagnosed in the last 2 years, and now it's the last 4 years, right?
Yes, that is right. And that's specifically important for the AL population, where some of the AL patients who respond exceptionally well to standard of care, their amyloid can go away. So we want to ensure that everybody who has a diagnosis of cardiac amyloid has cardiac amyloid when they come to Knoxville. So it's for that reason, not for any other reason.
Another question for you, Dr. Wall. part 2 of question on Dr. Wall's study. If patient has recently come out of remission, can they have already started treatment again and still participate, or does it need to be before treatment begins?
No, there's data. I can send you the data for clinicaltrials.gov, but we do need some clinical evidence of cardiac involvement. So the first step of the study is that we ask the patient to allow us to request their amyloid-related medical records, so we can check things like NT-proBNP and things like that. So to show there's cardiac involvement at the time recently, and so then that's one of the things we need to look for. But
Okay.
we have nothing to do with newly diagnosed or refractory or anything like that, with no criteria around that.
Okay, and John, question, this patient has been on the TALOR 101 trial for 2 years. Their troponin is now stable and their NT-proBNP is stable. They're thinking of leaving the trial because of that. What would you say to someone like that?
The other thing I'd throw in is, so the patient who's on the trial, they don't know if they got placebo or the real drug. And unless their disease, their organs are absolutely perfect at this point, if perhaps maybe they weren't getting the real drug. So there's the opportunity, there's going to be an extension with a crossover. And so then they would have the opportunity to get the drug, and maybe, it would make the organs that much better. So there's a lot we don't know and understand yet. So it's probably a good idea to stay on.
We could go on probably all day with this, but Dr. Dispenzieri, we want to take up some of our more treatments on our chart. We haven't touched on the IMiDs, we haven't touched on venetoclax, and of course, the big question of stem cell transplant. So we'll get back on Q&A, but we do want to hear from you about that, okay?
Sounds good. So I added just 1 or 2 slides on CAR-T when it didn't get covered. So hopefully we can cover that as well. So this is just another version of the same thing. As Muriel said, we've sort of talked a lot about the fibril-directed agents, but everything to the left of it is really going through how do you kill plasma cells? How do you shut down the manufacturer? And so again, just as a reminder, here's a patient, their bone marrow is where those plasma cells are made. And if there's a lot, we call them myeloma cells. They make immunoglobulins that circulate in the bloodstream, free light chains circulate, and they can form these fibrils that can deposit in the tissues and make patients sick. And so our treatment strategy as of present is to kill the plasma cells, and then hopefully the body can get better on its own. The newer drugs that we're talking about are, the KIL and NEOD, they're basically trying to facilitate that removal. And so I have a kind of a cartoon. This is the analogy I use with my patients is that plasma cells are making the light chain bricks to be laid into an amyloid wall. So you got like dozens of folks here making the amyloid, plasma cells, making the protein to make amyloid, build the wall. And then there's your body in the left. Your body's trying to take down the wall, but it's one against many. I can't keep up. But if you get rid of the guys at the other side, now your body can perhaps get the job done. And again, these antifibril antibodies are trying to make that even easier for your body. So what about transplant? Again, this is something that's been around for 30 years, in the amyloid space, where we, collect stem cells and give you high dose chemotherapy, give you your cells back. And what we know over the years is that you get complete hematologic response rates in about 40, if you don't have any treatment prior to the high dose melphalan, that's part of the transplant. About 40% of patients will get a complete hematologic response. More than 90% will have a hematologic response, but organ responses also, if you have that complete hematologic response, more close to 90% will have a renal response if they had their kidneys involved. And if you look here, if you had a complete hematologic response, again, more than 90% will have a cardiac response, liver response. And of course, if you look at patients who didn't get quite a complete response, but a VGPR, a very good partial response, even there, like 60% will have a renal response, and 70% will have a cardiac response. So it is good therapy, but it's hard work. And again, these data represent some of the best patients, because we know that if we take the sickest patients to transplant, we can really do harm, and actually they can die. Let's see. So Andromeda. This is Dara-Cyborg-D. This is the trial, very complicated slide, but in short, if you look at the light blue, VCD, which is bortezomib or Velcade, cyclophosphamide dex, it's done weekly for 6 months essentially, and stop and watch. Whereas the Dara arm, patients got the same VCD, but they also had Dara at that same time, and they continued the Dara for another 18 months. And what was found with that study is that if we look at hematologic CR, so the orange is the VCD without the Dara, and the purple is the Dara VCD. And the deeper the color is, the stronger the response. So about 53%, that's as good as transplant in terms of CR rates. You kill plasma cells very effectively with this regimen. And you can see then very good partial response in 25%. And what that amounts to in terms of organ response is, again, focusing on the purple, 42% of patients will have, and this is just at 6 months, a cardiac response, and a renal response at 6 months is 53%. There's a little bit more mature data and the numbers are a little higher. So this makes you say, do we really need to do transplant anymore? You can potentially do just as well, maybe without going through that big ordeal. And I think many of us in the field are saying, why don't we start with the Dara VCD, see how patients do, perhaps collect those seed cells for a rainy day. But if they get into a really, really deep response, perhaps we can just save those stem cells for a rainy day. Or perhaps maybe if they have more myeloma-like a disease, it just, there are lots and lots of those plasma cells, or maybe there's some things in the genes of the plasma cells that make us anxious, that we say, you know what, even though you've had a really deep response, hematologic response, I think it may not last. So why don't we go forward and just give that transplant? Again, there's a lot that we still have to learn. We don't have long-term follow-up in terms of overall survival or time to next therapy in this trial. So we're still learning. And this is, again, transplant. You can see that each colored curve is a different time period that patients were diagnosed. So the light blue, was 25 years ago when they were diagnosed, and the pinkish is people diagnosed, 20 to 15 years ago. And then the blue was sort of, people diagnosed from 2010 to 2014. And you see that patients are doing better and better all the time. And we know that there can be some really good durability. So that's where it's a conversation. It's not black and white. Transplant's not dead, but perhaps we don't have to do quite as much of it. So venetoclax and translocation 1114. So what in the world is translocation 1114 or T1114? So we all have, every cell that we have has chromosomes, 23 pairs, 2 of a kind, except for men who have, they have XY, whereas women have XX. But you basically have 2 copies of everything. And each of those little squigglies, are chromosomes. What happens with chromosome 11 and 14 is they sort of, one of the chromosomes 11 and one of the chromosomes 14 breaks apart, and then it heals back. But instead of healing with its proper partner, it heals with the chromosome 11 and 14 attached to each other, which basically drives the plasma cell. So that's actually happening in the bone marrow plasma cells. And so that when your doctor says, oh, you do or don't have translocation 1114, that's what they're referring to. And we can see that through a method called FISH, or fluorescent in situ hybridization. And on the left, you can see, it doesn't show that great, but that's a little green dot, and then here's a little red dots. The red dots are the chromosome 11, and here's the chromosome 14. Here is a fusion of green and red that gives you yellow. And so when we see that, we say, oh, you have a translocation 1114. And what's been discovered is that patients who have translocation 1114, they are very much, those plasma cells are very much susceptible to this drug called venetoclax, which we call a BCL2 inhibitor. And so this is like, one of the first reports of just, this is what we call a swimmer's plot. Patients were given venetoclax plus or minus dexamethasone, or with other chemotherapies like bortezomib, cyclophosphamide, et cetera. And what was found is that patients really were responding, and these responses could be durable. And so groups of physicians, getting together, looking at what we're seeing in patients with this 1114 with venetoclax, is that we're seeing that complete response rates in 41% of patients. And these are patients with relapse refractory. Now, some of these patients have not just venetoclax or just venetoclax and dex. They may have other drugs added in, but these are very, very impressive types of response rates. And in terms of overall survival, again, follow-up was only about 14 months, so not very long. But again, the survival is like 100% at a year or so, or almost 100%. And what we call progression-free survival is 90%. And so another look, this is something that is published out of Europe, where they looked at venetoclax. And again, these aren't necessarily patients on clinical trials. It's just they were able to get the drug, and they said, let's see how it goes. And so here's 51 patients. Median time from the diagnosis of amyloid to treatment was 24 months. And more than half of them had daratumumab. And so what you can see is what happened to each of these patients, why they discontinued. They would discontinue the therapy if patients had a very deep response, or obviously if somebody died, or if there were side effects or progression. But if you look to the right here, you can see the rates of complete hematologic response. Just with the venetoclax, 56% of patients had a hematologic complete response. Of those treated with venetoclax and dex, it was 60%. Venetoclax with bortezomib or Velcade, again, 67%. With Dara, 67%. And then 3 drugs, venetoclax, bortezomib, and Dara, 67%. So we have a lot to learn, but what we do know is that it's pretty darn good therapy. As far as toxicity, again, at this point, people look at the charts, it's retrospective, it's hard to know, how safe is it, but it appears to be pretty safe at this time. And so there are a number of clinical trials that are ongoing looking at this. There are 2 in China, and then they're relapse setting in the US, and that's just venetoclax and dex. And then there's another one with venetoclax, Dara, and dex. So side effects, so we have our groups of different therapies, and each of them has their really unique side effects. We heard a lot about the side effects of the bispecific and CAR T cells. As far as we didn't hear a lot about the fibril-directed agents, the reality is that they appear so far to be very well tolerated. Steroids, everybody hates because it causes all kinds of nasty side effects. And I have something on the next slide. But then, the alkylating agents, like you get the high-dose melphalan or low-dose melphalan, or the Cytoxan, that's part of that Dara-Cyborg-D. I'll show you on the next slide what those side effects are. And then the antibodies, they have their own different side effects. So if these little orangey things, that's supposed to be little snippets of the bone marrow, so myelotoxicity or damage to the bone marrow, all of these drugs, are hard on the bone marrow. in terms of the bortezomib, neuropathy, this is a little nerve, and also there is a little bit of cardiac toxicity. Ixazomib is another proteasome inhibitor. Carfilzomib is also a proteasome inhibitor, and it's a very good drug in patients with multiple myeloma, and it can be used in patients with AL, but it has a pretty significant cardiac toxicity, and so you really want to be very careful in using it and maybe not using it in a patient with cardiac, unless you're pushed to the wall and there's nothing left and you want to try something. But then, the edema that is part of the heart failure too. Dexamethasone can cause myopathy and swelling. Pomalidomide, so now we're moving into the IMiDs. Pomalidomide and lenalidomide can have some cardiac toxicity, and lenalidomide some nerve, peripheral neuropathy, and obviously bone marrow issues as well. Daratumumab can have a very little bit of nerve neuropathy, maybe a little bit of cardiac fluid retention issues. Venetoclax, again, some fluid retention and some potentially muscle stuff, but I haven't seen that in real world. Belantamab, ocular toxicity, and we're going to talk more about that in a moment. And as I said, we talked about the CAR T and T cell engagers. So moving to the IMiDs, these are drugs that are very important in multiple myeloma, and they are less important in amyloid, certainly not something we are using as a first-line treatment. They've been used mostly with pomalidomide or, with dexamethasone. Sometimes something like Cytoxan has been added in as well. You can add bortezomib in as well. But in general, as a single agent, the complete response rates aren't as high as you see, for example, than with daratumumab, or what you see with bortezomib or venetoclax. And there's just a lot of toxicity, and you have to just be very careful in using it. Not to say that you can't use it, but again, the Revlimid or lenalidomide, you want to start at a lower dose than you would in a myeloma patient. The pomalidomide is a little better tolerated than the lenalidomide. And so these are usually things that we save for, third line or so therapy. So belantamab, whoops, my name got in there. That's weird. Anyway, belantamab, this is a antibody, but it has this stuff here on it. And this is a chemical. This is MMAF. It's a chemical. And so we call this it's antibody, I'm sorry. There's basically chemotherapy attached to it. It's not a naked antibody. And so what that does is the antibody brings the, it attacks BCMA. And what it does is it brings this drug preferentially to the plasma cell. So you can not only have this sort of antibody killing, but you're also delivering poison locally. The problem with this drug is, and it was FDA approved for myeloma for a couple of years, but then it didn't meet its final endpoint. So it was pulled in the end of 2022. And then what happened was now there's been a study called the DREAM-7 study in myeloma that is a favorable study, so it may come back into the market in the US. So for that reason, I'm just going to show you a little bit of information. In Europe, it is still available, and the problem with this drug is it was used every 3 weeks, and people would get ocular or eye toxicity. They would get their sort of keratinization of their sort of cornea. A lot of times they wouldn't notice anything, but sometimes they would have blurry vision or difficulty with their vision. And so this drug was given, you had to see the eye doctor every 3 weeks when you got the therapy. And there was still eye toxicity because the doses that was started out were higher. So folks now are using it at a lower dose, less frequent. And so this is real world data from Europe, or actually the UK, where they were using a lower dose and every 6 weeks. Patients, the median time from diagnosis to using this drug was 65 months or over 5 years, 3 prior lines, 31 patients. Most are still continuing treatment, 11 discontinued, and mostly was because not a good enough response or progression. 1 was due to the eye problem, and 3 were for other reasons. But there were hematologic responses, with 35% of patients having a complete hematologic response and 23% having a very good partial response and 13% a partial response. So again, I think it is something. It wouldn't be my first choice for patients, but I think as we learn to use it better, it definitely is going to have value. Median time to next therapy was over 2 years, but 68% had some of this eye issue, but only 1 stopped because of the eye issue. This is another study. This is actually a real study being done in Europe. And they're using, again, sort of less frequent, using it every 6 weeks instead of every 3 weeks. Their response rates were not quite as high. And basically partial information, more people dropped out proportionally and more dropped out because of side effects. Okay, a word about CAR T, because I just added a last minute slide because Dr. Lentzsch didn't cover it in great detail. But this is chimeric antigen receptor T cells. So here on the slide, here is the myeloma cell, the big blob in the middle. And as she told you, BCMA is part of the decoration or coating on these myeloma cells. And she told you about how bispecific T cell engagers, could attack the myeloma through this, BCMA, and then these bispecific antibodies is sort of the same category. Antibody drug conjugates, that's that belantamab that I told you about. But here's CAR T. And so CAR T, basically what is done is your cells are taken and they're actually modified in the laboratory. So they have this fancy little receptor added to your own T cells so that it will better recognize this BCMA on the myeloma cells. And then it's given back to you after a little bit of chemotherapy. And basically, here is a study that was done in Israel that I alluded to in the Q&A before. They were patients that were actually quite sick. It's 9 patients. More than half of them had very severe cardiac disease, what we call New York Heart Association grade 3 or 4. And 4 of the patients had again, by the Mayo staging, grade 3 or A or 3B. But there were deep responses. So back to that minimal, residual disease CR, 6 of them, 6 out of 9 had deep hematologic responses. Another 2 had a very good partial response and 1 had a partial response. And the duration, so this is the really creepy part. 6 of these patients had died at a year, and the duration of response was only 4 months among the 6 who had died, and the duration of response in the other 3 is 19 months and going strong, 13 months going strong, and only when this data was presented, a half of a month and going on. And so we need to learn more about this in clinical trials and see. It's unclear why and how. They said that they died of, cardiac deaths. And again, if these patients had very, very, very frail hearts, it may just be that this was, quote, too late, but we still have a lot to learn. If there was a COVID death and cardiac. So again, here is our slide, all kinds of things that we can mix and match to treat patients. And trust me, we do a lot of mixing and matching. And again, not to, belabor, we have a site, msmart.org, where you can see kind of what we're doing at Mayo. But we sort of have algorithms that sort of guide, but things are very personalized, obviously. And with that, I'd like to thank you for your attention. And this is our folks.
Thank you so much. That was really great. If we could get either our chart up or Dr. Dispenzieri's chart up so we can continue with Q&A. And it makes it so much easier for our listeners if they have the chart to go by. That's great. Thank you so much. Okay.
And I neglected to mention there are CAR T that are FDA approved, and that was on my slide. There's ide-cel and cilta-cel. So, people may sort of put their toe in the water with a patient, an amyloid patient who has used up a lot of options. They could use those, but as Dr. Lentzsch was saying, and I agree, best done on a clinical trial so we really can understand the safety.
Thank you, thank you, thank you. Okay, let's get some more questions answered. Here we have some daratumumab questions. Okay, so on daratumumab every 4 to 5 weeks since about 2 years and in full remission, IV infusions last 3 hours plus. Could this time be shortened without any risk? I thought daratumumab was done as a shot,
It
but
can be done, yeah. So, I mean, the first approval was with IV, but then there's Darzalex or FastPro, which is just a shot, and people are in and out in, 15 minutes. So yes, it can be shortened. I'm not sure why the infusion is being used.
well, okay, we'll ask that person. I know that person. We'll ask her to email me and let me know why she's on infusions, and maybe we can figure out another answer for her after the, today, and we can get that question to you, Dr. Dispenzieri. Okay, here's another one. I've been on DARA for 5 years with anticipation of participation for the rest of my life monthly. Oh my goodness. Is this correct advice from my oncologist? The rest of their life, that's wow.
Yeah. So again, it's hard, I don't know your details and all the rest, but, the reality is amyloid most of the time is not myeloma. And, perhaps in a true myeloma patient that has proliferative disease, that would be the right strategy to, not give up and not stop because you're afraid what might happen. But in amyloid patients, the majority of them, shortened therapy is a very feasible thing. So, we're even doing a study looking for patients who are getting Andromeda-like treatment, if you get into a complete hematologic response, can you shorten the 2 years? There was in the relapse setting, the French had done a study where they, used very limited duration therapy. It was like 6 months, and, patients still had remissions lasting, on average 2 years. So again, there's things, it is different than myeloma, but there may be something particular about your disease that's driving your physician, to feel anxious and want to keep you on. I can't really speak to that.
Right. In the argument, stem cell transplant versus Daracibor-D, you say, well, people with Daracibor-D do real well, real quickly and go into wonderful same response rate. But when you look at the stem cell transplant, that 40% where they have a complete response, it's kind of a one and done situation. They go 5, 10, 15, sometimes 20 years before they might need another treatment.
Right.
And here you have Daracibor-D, which has great results, but they seem to be on it. The response maybe 2 years, then they have to go on something else again.
And the thing is, we really just don't know the answer. And but the transplant has its own side effects and toxicity and danger. And so everything is, you're always getting those scales out and trying to balance risk and benefit. And the important thing is to do shared decision making, right? I'll tell you what I know, all the information, that is available, but then it's up to the patient for their value judgments to how do they interpret all of that? And they like the idea of one and done. If they think, if I share that I think they're very low risk for, dying or severe toxicity with the transplant, maybe they say, yeah, I'd rather just get it over with, like Muriel was saying, and it's been around for a while and da-da-da. Or somebody else will say, hey, well, why should I put myself through that if I'm doing well? And, and I can do the transplant later. And both of those arguments are very valid, and there's not a right answer today. Maybe 5 years from now or 10 years from now, we'll have a right answer. But right now, we just don't.
Gotcha. Okay, the next one, another one where they're confused because they heard the ALX 2020 recruiting. Well, that's a TTR drug, so we're not going to answer that question. If you are a TTR and watching this, be sure and tune in March 9th. Okay, we already answered the question about can you take two from the same column, the Calum and the bertamimab, so we answered that. Any idea when Eliquis will be available in generic form. That's a cardiac drug, is it not, right?
It's a blood thinner.
Oh, okay.
Yeah, I honestly don't know. Yeah, it's a 10A inhibitor, and they've been around for a decade, you know, so, but I really don't know the answer. I'm sorry.
Does
Muriel,
anyone know- Yeah,
I can take a little bit of that. Eliquis has been added to the list of 10 drugs that, this is if you're on Medicare, that Medicare Part D, they have added a list of several drugs. Eliquis is one of them that the drug manufacturers are going to have to justify the reason that they have this drug in a higher non-tier category. They're going to have to justify that, but this is part of the Affordable Care Act, and it does not take effect until January of 2026.
Mm.
But Eliquis does have a foundation side. The drug company has a foundation side to help pay for Eliquis, but if I remember correctly, the threshold is fairly low for the amount of money you can earn each year, but they can go to the manufacturer's website and check on that and see if they would qualify for it. I don't know of any other assistance out there for that drug. I have looked because my husband is on it, and yes, it's expensive. It is
Okay.
not covered under HealthWell, and I just asked them earlier this week why it's not, and it is because it does not actively treat cardiomyopathy, so it's not covered under their cardiomyopathy fund, even though Entresto does. But Eliquis is not covered under any funds that I've found so far, and I've looked because I would like the assistance as well.
okay, thank you. Okay, okay, I've read that those with CD38 bright respond better to daratumumab. I have CD38 bright and responded quickly and still in complete response after 2.5 years. That's good.
No, patients, you know, again, we did, as Dr. Lentzsch had shared, we're typically not measuring like how bright or whatever. Patients seem to do well, but yeah, and patients, it can be a great drug for some patients. You know, patients are different. You know, we say you have the same disease, but everybody's disease is a little bit different, and some drugs work better for some than others. Some are fortunate that everything seems to work. And so it's a journey.
Okay. AL cardiac on CyBorD DARA, troponin comes down, but pro-BNP stays high consistently. What is the explanation?
So the NT-proBNP is basically a chemical, so to speak, that comes sort of from the heart when it's stretched. And so it's very sensitive to fluid overload, fluid retention. You know, you have a salty meal of some sort, and that can drive it up. And so often the dexamethasone, if you know, your doctors aren't working very carefully with diuretics, that can, you know, continue to be elevated. It's a very good sign that the troponin's going down, because that is something that is real, like tissue injury. That's literally little heart cells or cardiomyocytes dying. And so if the troponin's coming down, I think that's great news, but perhaps a little better supportive care in terms of, you know, maybe the diuretic management.
Okay. Patient completing 18 months of monthly DARA, would you recommend stopping DARA at 24 months or continue ongoing DARA indefinitely? You mentioned something about you were going to be doing a study on that at Mayo.
Yeah, we have a study where if patients get, you know, a CR, then we're sort of limiting the duration, and we'll see how it goes. Because again, based on the data in amyloid patients, it doesn't seem for the majority. And again, I can't speak to a case that I don't know the details. I don't want, you know, somebody to run home to their doctor and say they said stop it, you know, because there may be something very specific.
They will do that anyway.
Is driving, you know, the decision. But again, you know, if it's like an amyloid with fewer than 10% plasma cells, I don't think you have to go beyond the 24 months. I think that that's really adequate, especially if you're in a CR. You know, if you've only like had a partial response, well, you probably should have changed therapy by now and tried something else. And, you know, if you have a VGPR, it's kind of a toss of the coin. But again, they're just, how's your organ response doing and all these other things. But for most patients, I think 24 months, again, we don't have the data for sure, but I think that's plenty adequate. And unless they were extenuating circumstances, I'd feel pretty comfortable stopping at that point.
Okay, this next question. Well, this one is interesting because we do see several people on this combination. Do you ever combine venetoclax with bortezomib or DARA or both?
Right. And so as I showed you on that slide, it's like a mish, people are doing all sorts of things. And, I've had amazing experience just with the single agent. And so I tend to just, or maybe with a little bit of dexamethasone, but I start with that. And if I don't see a response, then I would look at adding something. And there will be a trial using sort of one using daratumumab with the drug, and then also people are using venetoclax with the drug. In the myeloma space, both of those combinations have been used and are being studied as we speak. But I always start because I've seen amazing responses just with the venetoclax and dex in the 11-14 patients.
Okay, but you are say there are some upcoming trials with venetoclax and DARA, or?
Yeah, so that one is, I showed clinicaltrials.gov there. I don't think it's accruing yet, but it's there. Let's see, I'm going to go back to my slide and see. So this is, venetoclax. So venetoclax DARA dex is called Altitude. It's run by Janssen. And I don't know 100% certainty if it's accruing or not. I don't think it is. And then there's a venetoclax dex that's ongoing in the US. And then there's a venetoclax with ixazomib, which is a proteasome inhibitor. So it's like Velcade. It's an all oral regimen with dex. So those are the studies that are sort of on clinicaltrials.gov. And then there's 2 other studies in China, which I don't think most of us in the US.
And a great reminder for our patients to go to clinicaltrials.gov, put in their search terms and see what's happening to stay up to date. We try to keep everyone up to date, but thank heavens there's lots of them going on right now, so which is really wonderful. This is kind of a vague one. What are your recommendations after, this isn't fair, after off medications for 2 years and come out of remission? We've been discussing all of these wonderful drugs in the arsenal, and I think if you go to a center where they know what's going on, maybe there's a good clinical trial for this person to go to.
Right. And if you go to msmart.org and look up amyloid, you'll see kind of what our algorithm is, depending on what you've been on and this, that, the other, and whether you're translocation 11-14. But if you are T11-14 and you've been off therapy for 2 years, I would strongly go to, if you can get on a venetoclax study or get compassionate use venetoclax, I just think that's a really great drug in that population.
Okay, venetoclax, you got it. Okay, for the AT01 based scan, it was shown to detect amyloid in the kidneys. 2 questions. Is it safe for those with kidney involvement? Does the drug not go to the kidneys like the other ones do? Okay, so these are the scans. So do we still have we do have Dr. Wall ready to answer. Look at that.
That's a great question. So the issue with imaging and renal insufficiency or renal problems, particularly with amyloid, is not normally the radio tracer. It's normally the contrast agent they give for the CT component of the scan. That can be toxic to the kidneys. And so when we did the imaging with 01 and the current study, we don't give CT contrast agent, which is very concentrated iodine, and it can be toxic for the kidney. So we had no evidence of acute toxicity in the kidneys from the imaging agent.
Thank you. Okay, this is an interesting question because we've seen this come up before too. And that is, okay, where to go? My husband was on venetoclax and it was working well. Because he had amyloid in his tongue, he could not swallow pills. So I was told to crush them and administer through his G-tube. Would crushing the medication reduce its efficacy?
You know, I'd have to ask the pharmacist that. I think if the pharmacist said it was okay, then I think it's okay. I'm gonna actually have to go in like 2 minutes because our grandkids are here and we've run over. So I'm sorry, like one more question.
We have two more.
Oh.
Okay, two more. So we're going to force you to stay for two more. Is it normal for cardiac symptoms, shortness of breath, fatigue with minimal exertion to worsen after starting darolutamide treatment?
It certainly can because of the dexamethasone and even sometimes the Velcade. But again, that's where daily weights are really important. Watching your diet, appropriate use of diuretics, making sure that you're on optimal management. Sometimes inexperienced cardiologists will give medicines, heart failure medicines like beta blockers, metoprolol, or calcium channel blockers, or ACE inhibitors or ARBs. And for many patients with amyloid, they do not tolerate these. So there may be some, medications that were added that may be making somebody feel less well, but certainly the dexamethasone, the fluid retention can be an issue. And so you need to work with your doctor on that.
Hey, Dr. Dispenzieri, I will ask you to please send me your slides. We have people that are very interested in a lot of your diagrams and graphs, and I want to share them. So please, please, please send me your slides as soon as you can so that I can let everyone have them. Okay. I just want to thank our panelists and Dr. Dispenzieri and Dr. Lentzsch, who had to leave us earlier. And if you mailed in questions, we didn't get to some of those, okay, but I promised you that we'll get answers to those and we'll email them back to you. And Bob will make sure that today's recordings appear on YouTube and the ASG website. Pharma usually doesn't allow us to reproduce their presentations, but if they do, we'll put them up. Otherwise, we'll just have to do without that. These will also be shared on One Amyloidosis Voice and in our Facebook groups. So we'll see you soon. Stay safe. Let us know if we can help you in any way. You've got our emails. We're always a phone call or an email away. And just call us day or night, preferably days. And don't forget the survey and stay safe, everyone. Thank you so much to all the people that joined us today.
Okay.
Thank you.
You.
Bye.